Welcome, everybody, to our next series on our symposium. Today is talking about immunology and machine learning. My name is Adeeb Rahman. I am the Senior Director of Single Cell Immunology here at Immunology, and I'm here with Jacques, our Chief Scientific Officer. I'm super excited about today's agenda. So for those of you who've tuned into previous parts of the series, I think one of the topics that came up in our last webinar was, in general, the utility of machine learning in immunology, but also how the immune system itself is just an ideal system for transfer learning because of the deep interconnectivity between all the different aspects of it, and the fact that there's shared features like gene expression that are shared across multiple different cells that can nevertheless result in highly cell type dependent attributes that connect to highly disease specific outcomes, but nevertheless have axes that connect back and forth throughout the entire system. And I think all of our speakers today have slightly different ways in which they're leveraging this interconnectivity to be able to gain insights that hopefully drive beyond kind of the historical knowledge that we have of the system. So without belaboring this any further, I'd like to go ahead and introduce our first speaker for the day, Sara Mostafavi, who is an associate professor at the Paul G. Allen School for Computer Science and Engineering at the University of Washington. And she's going to be talking about the deep learning of immune cell differentiation. Welcome, Sara. Looking forward to hearing you talk.
Thank you so much. I'm just going to share my screen, and then we can get started. Do you guys see my screen?
Yes.
Yeah, okay. And can you hear me okay? Yes?
Yes, we do.
Okay, awesome. Thank you so much for the invitation, Adeeb, Louise, and other organizers. I'm really excited to be here. So today I'm going to tell you a little bit about our recent work on trying to interpret deep neural networks in order to gain mechanistic biological insights, and in particular, how information about immune cell differentiation is encoded in the genome. But first, I want to briefly tell you a little bit about the research in my lab. So over the last few years, I've been really interested in questions that relate to genome interpretation. In one direction, which I'm actually not going to tell you about, we work a lot on developing algorithms for understanding how genetic variation across individuals impact various kinds of molecular or disease phenotypes. So some example of this work include models for identifying causal variants in rare disease patients, where we know any single rare disease patient has thousands of genomic loci where they differ from their parents, and we want to identify the smaller subset that are driving their disease. In the second direction, we've been really interested in developing models for understanding the propagated impact of genetic variant across different kinds of molecular phenotypes, including epigenomic, transcriptomic, metabolomic, and so on. And finally, we've also been really interested in developing models for try to jointly understand the impact of genetic variation and environmental exposures on disease phenotype. And here are kind of the computational twist to this problem has been to develop machine learning models that can infer unobserved and unmeasured environmental exposures from data that we can measure, like gene expression and epigenomic data. In the second direction, which I'm going to actually tell you about, we're really interested in understanding how the same genome, the single genome that we have in each individual, can give rise to so many cell types and tissue types. And as Adeeb mentioned, immune system is a really good fitting kind of tissue that we can do this investigation in. And what I'm going to tell you is based on our long-term collaboration with the Immunological Genome Project, or the ImGen project, and also Christophe Benoit's lab at Immunology at Harvard Medical School. And I'll point out to the key people that are involved in this project and the series of connected projects as we go along. Okay, so the basic motivation kind of goes back to this foundational question, which is how is it that the same genome can give rise to so many different cell types? And here's the cartoon example on the left. I'm showing a dozen or so immune cell types that we know are functionally distinct. This is among a cartoon of what's happening here, but there are at least 100 immune cell types that immunologists have worked out and have a good sense of their functional differences. And we want to understand how is it that all these cell types are differentiated from the same hematopoietic stem cells operating from the same genome. How does this kind of come about? So of course, main kind of key part of the answer is differential usage of regulatory regions across the genome. So again, here's a cartoon where we have 3 different immune cell types. This is the same genome, but each cell type essentially has its own distinct regulatory genome that it can use to control downstream gene expression. And putting it more formally, basically the main hypothesis for explaining this phenomena is that these different regulatory regions have differences in sequence composition that allow for representing different combinations of regulatory motifs that are bound by transcription factors and other regulatory factors that control gene expression. So even though at high level we kind of understand the principles for explaining cell type differentiations, we don't have enough details to really instantiate this hypothesis to be able to answer simple what and why questions as I framed them. So the what question would be, if I gave you a regulatory region and I asked you which cell type is this region regulatory for or important in, for any, you might not know the answer of this for many different regions. And second would be the why, which is a deeper question. So even if you knew a particular regulatory region is relevant to a particular set of cell types, why so? So what features, sequence features are hidden in that region that allow the cell to make the determination about cell type specificity of that regulatory regions? So in the last decade or two, there's been like a revolution, as you all know, in measurement technologies that we can now measure all sorts of functional output across the regulatory genome. So starting from ChIP-seq to DNA-seq, and more recently to ATAC-seq and many other assays that we can apply. And if we apply these systematically to a large number of cell types and tissue types, we can start to be able to answer the first question, which is essentially try to annotate all regions across the genome based on their cell type specificity. But what I want to talk about is the second question, which is the deeper why question. And in order to do this, the data by itself is not enough. We need models that can basically figure out what the logic of the sequence that allows the determination about cell type specificity. So, so for the rest of the talk, I'm going to tell you about our approach for doing this. And this is mainly relying on large-scale generation of ATAC-seq data. So for just sake of having the same consistent terminology, let me just go over a few terminology things. So in an ATAC-seq assay, what is typically done is for a population of cells of interest, we can, through sequencing, identify regions that are chromatin-free. So we're going to call these regions open chromatin regions. And if we find open chromatin regions in a population of cells, we're going to assume that these represent a set of regions that are likely to be regulatory active for that cell populations. And then similarly, when we're talking about a population of cells, we're going to have a quantitative signal for each region, which is essentially related to the proportion of those cells that have that region open or likely to be regulatory. So a few years ago, as this ATAC-seq assay became mature, it was possible to apply it systematically to a large number of cell types. And one kind of exciting project that took this direction forward was the ImGen project, a collaboration between Christophe Benoit, Jason Buenarrostro, many other immunology labs, and including myself on the computational side. So what all the brave immunologists did as part of ImGen was basically isolate 90 distinct immune cell types from adult mice. And this was really exciting because these 90 cell types covered almost all well-known and well-characterized immune cell types that are known to be functionally distinct. So the dendrogram that I'm showing you here, each node here is an immune cell type, and we know their differentiation distance from each other. And the colors represent the lineage that they fall into. So these are broader immune lineages. And what I want to highlight is that from each of these lineages, there's actually multiple subtypes that are represented. So in the first kind of pass of analyzing these data when it came out, what we did was really focus on answering the what question, to annotate the genome based on regulatory elements across the immune cell type. And essentially, we can summarize these data as about 500,000 peaks or open chromatin regions, OCRs, that have activity across any of these immune cell types that's been assayed. So now what I want to tell you about is how we use the same data to go a little bit further and answer this why question. To remind you, what we want to know is that given the sequence composition by its own, can we figure out what's the logic for that piece of sequence, that regulatory sequence, to be active in some cell types and not others? So for this, the way we view this problem is through the lens of supervised machine learning. So what you can imagine is that we would like to train a machine learning model that can take as input a sequence that underlies each of these OCRs that we find. So these are typically about 200 base pair long sequences. And on the basis of sequence content, can use multitasking or transfer learning, however you like to phrase it, to predict regulatory activity according to the ATAC-seq assay across all the different cell populations that we've measured. So when we frame the problem in this way, of course, the goal is not just to make prediction, like in sample type of predictions, but we use that prediction accuracy as a proxy to actually answer two deeper kinds of questions. So the first thing that we might be interested in once we have such a model is to try to interpret it and learn what did the model learn about the relationship between sequence and regulatory activity, and can this give us some clue into how cellular differentiation actually works and how is it encoded in the genome. And the second thing that we might be interested in, and this relates to the kind of the first direction of research in my lab that I initially mentioned, is to try to figure out the effect of unseen mutations on regulatory function. So I can do hypothetical in silico experiments and say, well, what if I change, a C to a G or a T to a C? How would that change the prediction output in terms of regulatory activity of that region across the many immune cell types that I've measured? So, as you might infer, is that for this model, we're going to use a deep neural network. And your first question might be, why do you want to use a deep neural network that's going to make interpretation and everything else kind of complicated? Why don't we just use a simpler model and solve this interpretation problem easier? For that, I'm just going to reference three of my favorite papers that really inspired our work. And essentially, without going through the details, since a few years ago, what people have been really able to show is that once there's sufficient data for building models that go from genomic sequence to some sort of functional prediction, having nonlinearity and complexity to be able to infer features in an unbiased way is really important for getting accurate predictions. And so this has been already demonstrated in a few different kinds of problem that takes genomic sequence as input. So we know that this being able to extract nonlinearity and kind of an unbiased representation of features is really important. So given this previous work, we had two goals here. One was that, can we really push the prediction boundaries in terms of predicting granular cell type activity or regulatory from sequence information, where the work that was done prior was mostly focusing on tissue level differences, which are a little bit easier to spot with various models. And the second question that we had, or goal that we had, was can we actually interpret these complex models in a robust way to learn about biological mechanisms? So for this, we developed a model called that we call AI-TAC, and this was a really close collaboration with Christophe Benoit's lab, postdoc from their lab, Ricardo, and two students from my lab, Mark Ma, that was actually at the time undergrad, a brilliant undergrad student, and now he's doing his PhD, and Sasha Maslova, a PhD student from my lab. So what the AI-TAC model does is takes as input these 200 base pair sequences underlying the ATAC-seq region. So there's about 500,000 of them, as I mentioned. And we trained this convolutional neural net model to predict jointly the intensity, the measured ATAC intensity across the 90 different cell types that's been assay by the ImGen consortium. And so this model is a multitask learning model. So it has a bunch of shared layers here, where the union of features that are important for all the prediction of all these different cell types is learned. And this last layer is where the information becomes specific for predicting activity in any particular one cell type. So what I wanted to also mention here is that so we fiddled around a lot with how are we going to design the architecture of this model, and what we realized is that actually some of the previously published architectures in terms of number of nodes and in terms of number of layers tend to work well on our data set too, which was never modeled in this way before. So there seem to be some sort of architectures that fit general data set with a given structure in regulatory genomics, and in particular, from going from sequence to some sort of functional output prediction. But what we thought was, realized that what was a lot more important is the loss function that we use to train the model. And so let me tell you why that's actually an important consideration. So one challenge that we have with data sets like this, when we want to go to cell type resolution, is that computationally, there's actually a big class imbalance problem. And what I mean by that is, if you look at two neighboring cell types here in this differentiation tree, so for example, I'm showing you data from T-naive cells and T-regulatory cells, which are kind of close together. They're both subtype of T cells. If you look at regulatory regions and the intensity of those regions across the two cell types, you see that it's pretty correlated. So there's a small number of regions where that are specific to each cell types. So that means that if you don't focus your model, or you don't emphasize for your model to learn from these kind of small number of examples that are cell type specific, you're just going to learn the shared logic of regulatory activity across cell types, but not anything cell type specific. And the way we solve this problem is to use just a simple correlation loss. So instead of measuring what's standardly done with this kind of regression problem is, you would use mean squared error, but instead of using mean squared error, we're just measuring the correlation between predicted and observed outcome during training, and we use 1 minus that correlation as the loss function for our model. And the reason why this helps is that through this correlation loss, we can decouple the effect of signal intensity from kind of which, so we can de-emphasize signal intensity when we learn how to predict different OCRs. So we trained this model, and the first thing that we did, as is standardly done, is to use cross-validation to assess its performance. So we can do this through leaving out some portion of the OCRs that we have as our for our test set, and then measure based on observed data how well we can make predictions on our test OCRs. And if we do this, so this is, and remember that we use a correlation as our loss. So what we can do is also measure for each OCR, our model is going to make a prediction, which is a vector of length 90, and that has the regulatory activity for each of the cell types that we want to predict. So we can correlate that vector of like 90 with actual observed ATAC signal. And if we do that, we get a distribution of correlation values, which is this orange curve here. And then we can assess, how good is this? One way to assess is by creating a null distribution. And for us, in this case, what we did was shuffle our sequences, fed them back to the model, made prediction, and see how well, what's the correlation on this null set of OCRs that have been used as input to the model. And when you do this as expected, you should get correlation distribution that is centered at 0. And based on this, you can quantify the statistical significance of your prediction. So one thing that was started to get really exciting is that for some of these well-predicted OCRs, if you actually look at the prediction versus ground truth, we see that there's a lot of granularity. So within the T-cell population, this is example prediction for one OCR, there's a lot of variability in terms of which T cells, subset of T cells this OCR has regulatory activity in, and you can see that the predictions are matching the ground truth. So kind of cross-validation is never end of the story, as you all know. So we really want to test this model using an independent data set. So what we got lucky with was that there was another study. So if you remember, ImGen was based on data from mouse. There was another study that was published a few years ago with human ATAC-seq data done in a somewhat of a similar way across many of the cell types that were shared between mouse and humans that were both measured. So we just took our trained models and applied it directly to this data from human. And again, we saw that the models can definitely make predictions beyond what would be expected. So this got us really excited to try to ask, so, okay, now what did the neural networks or our models have learned about the relationship between sequence and regulatory activity, and how do we gain insight by analyzing their parameters? And to do this, as you may know, there is now hundreds or so different interpretation methods for trying to figure out what neural networks have learned. And conceptually, you can think of them as falling, and all these interpretation methods falling into three different categories. Methods that work based on optimization or activation maximization. Methods that work on feature attribution, where for any given prediction, you can find the features that were important for that particular predictions. Or a simplest approach, which is kind of node activation and ablation. And for the sake of time, that's what I'm going to briefly tell you about. So what we did was take this simple approach where for each node in our neural network, we're going to try to figure out what is the motif that is learned that allow it to make these predictions, and how important is that motif. So our step basically has three parts to it. In the first part, for each node in the first layer, we're going to ask what are the sequences that activate that node, and that's how we extract a motif that filter or that node had learned. In the second step, we're going to remove that node from the model and measure how much error that's causing in our prediction. So that's a way to get at the importance of that motif. And the third step, which was kind of, this is something that we came up with and realized this is actually a really important step, is we're going to repeat this process 10 times in an ensemble approach. So what I mean by that is that we're going to train 10 separate models and repeat this whole process and see how stable are the influence and the motifs that we're finding. And this gives us a sense of kind of replication across actual parameters that are learned by the models. And so for the sake of the time, maybe I'll just summarize that. I think one important takeaway that we had was if we take into account this reproducibility metric, so for each motif that we've learned, if we know how reproducible that motif is across different trained models, then we can use that information to actually find motifs that are much more stable in terms of varying interpretability methods being able to find the same motifs. So another way of putting it is that once we account for uncertainty in parameters, different interpretability methods actually give you the same thing. It's just that parameters that are not very stable, those result in method-specific motifs. So on the biology side, one thing that was really cool was that for each of the nodes in the first layer of our network that we had, we annotated each filter with a motif, and for that motif, we went and checked databases, transcription factor databases, to see if that corresponds to a motif for known transcription factors. And when we did that, we realized that most of the nodes that were learned are actually representing known immune transcription factors, which is really exciting because this information was just learned by the model by itself without any kind of intervention in terms of feature engineering. So I mentioned most of the motifs were actually known, but there were some that are stable and influential and unknown, and those are some of the ones that we've been looking into. And this In closing, what I wanted to mention is that so I only told you about kind of scratch of the surface of interpretation. I told you about looking into first layer filters, but all the exciting stuff in this deeper layers of the network where combinations of motifs are learned. And one way to kind of understand how many motifs are important for prediction of any particular regulatory region, one way to do this is that you can quantify per OCR that you predicted how many motifs were used for that prediction. And that's a way to get at how complex is the regulatory code embedded in these OCR sequences. And if you do this, this is basically the histogram that you get. So on average, you need 2 or 3 motifs to make accurate prediction, but there are many OCRs that you need much more than that. And there's very combinatorial rules that specify cell type differentiation. So for that, I'll close with a few discussion points and tell you about where we're going with this. So we've done a lot of work in trying to extract interaction and nonlinear effects. And this is kind of, I would say, a very active area for method development because it's really hard to do that in a statistically rigorous way. And there are some biological reasons for this, is because any given combination of motifs is seen very few times, even across all the immune cells that we have. So that basically results in a hard statistical challenge in figuring out which ones are robust and stable. And in terms of our future work, we're looking into kind of adopting these kind of models for single cell ataxies and much larger number of cell substates and types. And we're also developing models for cross-species inference that include both human and mouse. For that, I'll just conclude by thanking all the co-authors on kind of this work that I mentioned, in particular Christophe Benoit's lab, Ricardo Ramirez, and to all my students, Sasha and Mark. And thank you so much. Sorry if I run over by 1 or 2 minutes.
Thank you so much, Sara. All right, so that was terrific. I do want to encourage all of our viewers to submit questions, some of which we can pose to the speakers directly, and some of which we can save and reserve for a session at the end for our moderated discussion. I guess at this point, it looks like we don't have any questions that have come in thus far. So we can move on to our next speaker. Unless, actually, let me just confirm that there are no questions that have come in. There are not. All right. So next up, we're going to move on to Samantha Riesenfeld. So Samantha is an assistant professor at the University of Chicago. She's part of the Pritzker School of Molecular Engineering and the Department of Medicine, and also the Institute for Biophysical Dynamics. She's going to be talking about modeling continuous and dynamic immune cell states. Samantha, I'll let you take it away.
Hi, thanks. All right, I'm going to try this again. Let's see how it works. Okay. Can somebody send me a sign if you don't see my slides or hear me very well? This is a new format for me. All right, so I'm going to assume that I'm talking audibly and that you can see what I'm talking about. Thank you so much to the organizers for inviting me to participate in this symposium. I really appreciate that these symposiums that you've been hosting are both really stimulating and also short enough to fit into a busy day. So I'm excited to be here today and fit into your busy day. Thank you. My lab at the Pritzker School of Molecular Engineering at the University of Chicago works on developing and using machine learning approaches that leverage new types of large-scale genomic data to understand complex systems, especially in the context of immunology. In this talk, I'm going to focus on what will probably seem relative to Sara and Rahul's talks in a relatively simple setting, at least by the standards of large-scale machine learning systems like the one Sara was describing. And that's because I think it highlights some problems that are still actually challenging for us to even identify, much less solve in an unsupervised way, and that are really relevant for immune studies. So I'm going to focus on continuous cellular phenotypes in immunology, especially those that arise outside standard contexts of cellular differentiation and development. It's common outside those contexts to refer to cell types and phenotypes as essentially discrete categories. The idea that cells might exhibit a continuous range of phenotypes is actually becoming increasingly relevant. And that's partially due to the ability for us to actually see these kind of mixed phenotypes using single cell RNA sequencing and other developments in sequencing technologies. So a beautiful example comes from the labs of Itai Tirosh and Maria Suva, who found, this is from a paper a couple of years ago, where they found that cells in glioblastoma tumors range across a broad transcriptional spectrum delineated by established cell type phenotypes. So the extremes are phenotypes like astrocytes or null progenitors, but they found this kind of range across the intermediate space. So you can abstractly think about defining this transcriptional space using coordinate axes that define the degree to which a cell or a tumor exhibits each cell type specific phenotype. So this is really a 4-dimensional representation here, but I'm trying to show it on a 2-dimensional slide. So the coordinates might not quite make sense, but they encode a range of phenotypes that are measured originally in thousands of dimensions. And it suggests that we might think of glioblastoma as having an underlying continuous landscape, right, through which cells or tumors evolve over time and in response to treatment, changing the degree to which they exhibit cell type specific phenotypes. So that characterization of an individual cell as a sort of hybrid monster is kind of natural in the context of cancer. Because cancer involves cells being reprogrammed to behave in very non-standard ways. It sort of looks like a monster anyway. But I think this is an idea that's becoming more relevant in physiological contexts as well. And that includes immunology, where cell type models are traditionally very discrete. They're based on the expression of characteristic transcription factors and cytokines. So an example that might be familiar to some of you at least is the essential Th1 versus Th2 model that was established about 20 years ago. And it describes how cues from immune cells in the environment determine whether a naive T cell differentiates into a Th1 cell, which fights intracellular pathogens, or a Th2 cell, which helps expel extracellular pathogens and repair damaged tissue. But over time, this framework has gotten increasingly complicated, and that reflects the discovery of additional types of T helper cells with different functions, and evidence that these T cells don't always behave like terminally differentiated developmental lineages. So under the right signals, they can transition from one type to another. Intermediate T cell states have also been identified even more recently, sort of where the function is actually in that intermediate state. And they appear to combine traditional polarized cell phenotypes. And that picture starts to resemble the one that I just showed you in the cancer context. So these developments all hint at the potential continuum underlying T helper cells. And that's a notion that we'll see applies to other immune cells as well. So here, now I'm going to sort of talk about how machine learning techniques can hide or help reveal the existence of intermediate phenotypes and continuous trajectories among, in this case, innate lymphoid cells or ILCs, and why this matters. So ILCs are a recently discovered subset of innate immune cells. They reside in barrier tissue like the skin, and they're typically categorized into 3 main subsets that are analogous actually to 3 T helper subsets. So the conceptual model here is essentially that there's a specific cell type that's ready and waiting to meet each type of threat. And so I'm sure you're aware after the last 2 years that immune responses, when they become dysregulated, can also lead to pathologies themselves, right? And so one example is psoriasis, which is an inflammatory skin disease. So in this story, ILC3s are implicated in driving skin inflammation during psoriasis. And the problem for us really started with the fact that rather than sort of hanging around in the skin ready to fight, the ILC3s seem to appear out of nowhere. And that view is actually reinforced, you'll see, by kind of a standard analysis and visualization. But our analysis and experimental validations reveal that actually at least some of these ILC3-like cells arise from other ILCs. And in fact, there's this whole spectrum of ILC phenotypes that's much larger than what's suggested by that simple model on the left. And so conceptually, rather than requiring ILC3s already in place, this inflammatory process could be linked to a broad range of ILCs that have some varying, possibly stochastic potential to respond to inflammatory signals by transitioning to and converging on this pathogenic ILC3-like state. So you could imagine that would really change your understanding of what's driving disease. Okay, so I'm just going to go, one more biological slide to give you just a little bit more detail that will help maybe clarify later points. So the basic model originally is that there's a trigger that causes dendritic cells, a type of immune cell in the skin, to send inflammatory signals to T cells. And these respond by producing other inflammatory cytokines. And those cytokines drive the epithelial keratinocytes in the skin to proliferate. And the skin thickens and you get these typical lesions. And it turns out that, as I said, ILC3s also appear in lesions during clinical psoriasis. And they respond, they're sort of analogous to these Th17 cells. They respond to the same IL-23 inflammatory signal that's sent by dendritic cells in the same way that the T cells do. And as early responders, they can also amplify inflammatory signals for the T cells. So the question arises from the fact that in steady state skin, the predominant ILC type that was reportedly observed were ILC2s. And those produce a different set of cytokines. And so, we wondered where these ILC3s were coming from. And so there are experiments and analyses that we did that I won't have time to describe that suggest that neither recruitment of circulatory cells nor proliferation, while both are possible, they probably don't explain at least not fully, the rapid increase in ILC3s that we see. So we focused on this question of whether ILC3-like cells might be arising from other skin ILCs. Okay, so we used this mouse model of disease where the IL-23 inflammatory signaling cytokine is injected into the ear skin of a mouse every day for 4 days. And that causes that same rapid increase in skin thickness, which is analogous to a true psoriasis lesion. So to analyze how the skin resident ILC population changes during this initiation of disease, we took tissue samples of the ear tissue from biological mouse replicates at each day of the experiment, and isolated ILCs from these samples and performed single-cell RNA sequencing on the ILCs. So most of you probably already know that this results in a large sparse matrix of transcript counts that encodes the noisily measured activity levels of thousands of genes in each of thousands of cells. So I'm giving you an overview of that data now. And this is a nonlinear two-dimensional embedding. And each point here represents a cell that's colored either blue if the cell was collected at the indicated time point from the label, or gray otherwise. And one thing you can see right away is that there's this specific region that I've circled, which is unpopulated at day 0 and then becomes densely populated by day 3. And this embedding is actually a force-directed layout, or what I'll call FDL, of the top diffusion components of a K nearest neighbors graph, and that's constructed from the residuals of performing a variance stabilizing normalization of the gene counts. So that's a mouthful, but I think the key point here is that spectral embeddings like diffusion maps tend to emphasize connections between different parts of the data, especially narrow or sparse paths that connect different regions, because they tend to overestimate their lengths. So I like to envision this data. Maybe I naturally think in high dimensions, but I think of it as a high dimensional body with sort of tentacles sticking out, sort of like an octopus. And that's apparently also a structure that arises naturally in self-organizing social networks. And those tentacles represent ILC states with somewhat distinctive gene features, but there's a lot of continuous variation there. So I want to just show you that that structure is much less apparent if we use a more standard visualization pipeline with the same data. So, you know, even if you change some of the parameters, I mean, here I've selected slightly different ones, but the important thing here is that we've built a UMAP, a uniform manifold approximation projection embedding of nearest neighbors graph from the principal components of the log normalized corrected counts. And you can change several of those parameters and you'll still get roughly the same picture, which is the main difference being that the ILC3s are very separated, right? And some of the tentacles are also kind of smoothed out. And so that reflects the tendency of UMAP to split apart high density regions of data that are connected by low density regions, which is actually desirable when you're trying to visualize clustering analysis, but it's less useful for highlighting connections, especially sort of sparse connections. And that separation of the ILC3s from the other ILCs also reflects the fact that in this data, the top global PCs mainly capture differences between the ILC3s and the other ILCs rather than axes of similarity. In any case, it's clear from this big cloud / octopus body that there's a broad continuum of phenotypes among the ILCs, even at steady state, before ILC3s and inflammation are induced. So we wanted to analyze this space in a way that could at least represent and capture continuous phenotypes. So not using a kind of typical clustering approach. And we use probabilistic topic modeling, which is also, depending on your machine learning circle, it's known as latent Dirichlet allocation or grade of membership modeling. It results in a type of non-negative matrix factorization. And it might be familiar. It's very well established in text mining and population genetics. So in this model, the data is represented in terms of a small number of latent gene programs or topics. We use this sort of text mining terminology, where each topic is represented as a distribution over genes without, each cell is represented as a distribution of weights over those topics. So the cells become suddenly represented in a low k-dimensional embedding. And the gene loadings and topic weights are inferred by fitting a probabilistic model without any knowledge of known cell types or gene pathways. And you can we assessed models fit across all of these cells for a range of choices for the parameter of the number of topics, and eventually selected one with 17 topics. Within that range, it didn't change immensely what the picture was. So as Sara, mentioned, there are lots of alternative approaches we could have used, including other Bayesian or non-Bayesian latent factor models and deep learning type architectures like variational autoencoders. We used this approach mainly because it was simple and it gave us immediately interpretable and useful results. It definitely has drawbacks. We're not wed to it, but I think what's more important than the choice of any specific approach in this kind of setting is really if and how the results can be leveraged downstream to understand the data. So I'm going to show a little bit about how we did this in this context, but I think that's actually, trying to know what to do with these kind of representations. It is right now really art, and I think there's room for development there too. So let me show you some of the results just to give you a sense of them. One of the inferred topics captures this dominant program. One of these inferred topics captures this program that's the pathogenic pro-inflammatory program that isn't present at day 0 and becomes, increasingly present and is really dominant by day 3 in among a subset of the cells. So in this plot, I've colored the cells by the weight of the topics. You can see, that those cells that are in that region that arises later in the inflammatory induction process are high for ILC3-like topic, And then here we've scored the genes. These aren't actually exactly the gene weights, but they're it's kind of a way of scoring the genes for how specific they are to a topic, how well they distinguish a topic from another one. And so they're ranked there. And we see a lot of genes that are very closely associated with ILC3 cells. So, and we also, check, you can see the sort of expression of specific genes, this inflammatory cytokine IL-17 and the receptor for the IL-23 cytokine. All right, so just a couple of other programs. We saw one that's associated with the expression of ILC2 associated genes. And as I mentioned, some studies suggested that ILC2s represent the entire spectrum of the ILC population in the skin. But in our study, these sort of type 2 cytokine producing ILCs that express characteristic genes, including this gene IL-1 RL1, which is a receptor for IL-33, another cytokine, compose just a fraction of the steady state skin ILC population. And so contributing to that population is a set of cells that are high for this other program that we identified, which feature genes like the transcription factor KLF2 that are associated with T cell quiescence. And so we nicknamed this topic quiescent-like. Cells that are high in this topic or in many of the other topics just fall outside the established profiles for skin resident ILCs. So I'm not going to go into all the topics. I just want to at least show you kind of what it might look like. Some feature genes with known immunological relevance. And then there are a bunch of topics, which I've kind of splashed at the bottom, that feature genes that are related to fundamental cellular functions. And in most single cell studies, we would attribute those to technical or biological noise. But in immune cell studies, they can also stem from relevant biological signals. I am sorry if you can hear my dog. He's supposed to be being watched by someone. So anyway, during activation and response, immune cells change profoundly in size and proliferative state and metabolic state. So you can get similar kinds of features that are actually really confounded with important biological signal. And without further experiments, it's kind of difficult to hypothesize the significance of these topics. But we focused here mainly on, rather than sort of using the whole breakdown, we focused on using topics that are clearly linked to the ILC2-like, ILC3-like, or so-called quiescent state. So here I'm showing plots where the cells are colored by their weight for topic 3 if they're from that specific time point, or gray if they're from a different time point. And you can see actually that as early as day 1, you see an increase in cells that are high in that pro-inflammatory ILC3-like topic. Those are circled there. I hope you can see it. It's not too small. But some of these cells also have high weights in other topics, such as topic 2. And that pattern suggests the possibility that the earliest induced ILC3-like cells originate from the ILC2-like state. And similarly, some ILC3-like cells that arise a bit later by day 2 have relatively high weights for topic 1, that quiescent-like state. And so in general, we see this trend that it suggests that ILC3-like cells might originate from multiple states. And we wanted to reconstruct these trajectories to try to understand and validate the changes that cells might go through to arrive at that state. So we use this method that's based on optimal transport, which sequentially predicts the cells from each day that are most likely to have given rise to the ILC3-like cells at day 4, depending on kind of a cost for matching probability distributions. And this analysis predicted potential ancestors that spread this broad set of cells across the continuum. And that's consistent with the idea that we saw using the topic modeling that ILC3-like cells might arise from multiple origins. So to infer potential trajectories of cell transitions, we start with pseudotime inference, which you might have heard of. That's, in this context, pseudotime refers to the order of a biological process. And it leverages the idea, estimation of pseudotime leverages the idea that cells that are at proximal stages of the process are more likely to have similar profiles than cells that are at distant stages. So even if we don't know exactly where they were, by using their distances, we can try to recreate this ordering. But that estimation doesn't account for complicated processes that involve branching or converging or oscillations. So, in some cases, pseudotime is really looks like a function of physical time. So for developmental or differentiation processes, like the example I'm showing here from early zebrafish embryogenesis, it's common to combine pseudotime estimation with branching estimation and fit a tree-like structure to model cell fate trajectories. But that's not the case here, really. Well, it is in some ways, but not in others. So for one thing, a single time point can contain a distribution of cells across many different stages of immune-related processes. So that broad spectrum of steady state phenotypes that we observe could potentially be a result of cells being distributed across stages of steady state processes. And another challenge in this context of the ILC's data is that a standard tree isn't the right model for representing the cell state transitions. And finally, because the transitions are really rapid and relatively sparsely populated by captured cells, they're not easy to identify or visualize using a lot of standard methods. So we inferred potential trajectories using a biased diffusion process between starting and ending subsets of cells that we selected using a combination of diffusion maps and the topic modeling and the optimal transport. And so these cells had extremal values for topics 1, 2, and 3. And there was one other topic sort of in the cloud. Because we have real time points, we could infer directionality for some of the trajectories, at least the ones involving ILC3-like cells, right? So we can, once we know which cells, the ordering of the cells, we can also see if that ordering more or less corresponds to their the time point in which they were collected, and get a sense of directionality that way. But we'd like to be able to kind of understand directionality when we don't necessarily have that information as well in other contexts. So one of the important trajectories that we identified is this one that I'm showing, that I'm focusing on here, which was from ILC2-like to ILC3-like cells. And we were able to use single cell ATAC-seq to validate that there was this potential for ILC3-like expression at the level of the chromatin. So as Sarah was describing, the chromatin contains a lot of information about the capacity of cells to express different gene programs associated with distinct cell types. And single cell ATAC-seq measures noisily which genes in the genome are accessible. So here we can see that, there's a lot of activity in IL-5, which is an ILC2 associated cytokine, both before and after treatment. The after treatment is from day 4, and that's in the pink on the left. And there's most of the type 3 gene for here I'm using as a representative this cytokine receptor IL-23R. It's only after induction. But what's interesting is that the transcription factor ROR gamma, which is associated with ILC3s, is actually present both before and after, and in cells that aren't expressing any of the type 3 associated cytokines, but are expressing or potentially expressing type 2 associated cytokines. And we see something similar for this quiescence associated transcription factor KLF4. So I'm just about out of time, I think, but I just want to take 1 minute to say that we actually did a lot of validations of these trajectories. We used in vitro methods, but sort of the best validation is using a fate tracing mouse, where we actually could track whether cells that ever turned on this very quintessential type 2 cytokine IL-5 then later turned on a type 3 cytokine IL-17 or IL-22. And we saw that indeed, like, there are cells that express IL-5 and then they stop expressing IL-5 and start expressing these other cytokines. And so that really validates that at least some cells are going through this transition. This is a difficult assay, and so we lose a lot of cells. So we don't really know the extent to which cells are going through this, but at say at least 10% of them are or something. And that happens even more when there's a secondary challenge later. Okay, so I just want to say very briefly that for the, in terms of sort of the larger challenges and looking forward, this experience really made me think a lot about how we might be able to reveal the possibility of these transitions in a less supervised way, right? Because here we knew a lot about the system and we had specific reasons for suspecting that these transitions existed. But obviously we might not always have that. So one thing that we're looking at is better techniques for estimating and using RNA velocity. There's been really great work on how to leverage information from unspliced counts to infer transitions and their directionality, but they don't work very well in this context. Otherwise, we would have used it. And so trying to understand why and how we can improve that. Also, techniques that better leverage local information to infer relationships. So this global PCA is, can be very uninformative or even misleading about local relationships and transitions. And also, I'm interested in approaches that explore and visualize relationships that otherwise aren't revealed using sort of current analysis and visualization pipelines. There's a lot happening in high dimensions that we can't see when we try to force everything into 2 dimensions. Okay, I will skip the summary because I think I'm out of time, but I just want to thank my collaborators. The lead on all of the ILC's project in the experimental side is Piotr Bielecki, who used to be a postdoc and is now in biotech in Richard Flavel's lab. And I had great computational collaborators, JC Hutter and Elena Torlai-Trillia in the formerly in the Regev lab at the Broad. And I'm also really appreciative of my current group, which is just 2 years old, because they're really pushing forward some exciting ideas that spring out of this project. And I hope I'll be able to talk to you about that at some point in the future. Okay, thank you very much. And I'm happy to take questions, I guess, later if we're out of time now.
Thanks so much, Samantha. So I guess we do have a couple of questions, one of which, so Paul asked a question I'm actually going to save for our moderated discussion at the end, but 2 that are pretty directly relevant to your talk. One from Brian Lee at the Icahn School of Medicine at Mount Sinai, who asked, do you also see ILC3s circulating in blood? And if so, how similar or not is the phenotype to those that you see in the skin?
That is a great question. There have been sort of parabiosis type studies to see the extent to which there is circulation. We didn't do that because they were sort of done, and because and we didn't actually measure blood. I mean, in general, ILCs are incredibly rare, and they're very, very, very rare to see in the blood. So you'd have to sort of kill a lot of mice to do that, I think. But it would be interesting to think about how one might do it. There have been some cool studies. There was one out of Ron Germain's lab that showed that in some cases it looks like ILC3s, or maybe that was ILC2s. They're sort of ILC3 like ILC2s, whatever. They transit during lung inflammation from the gut to the lung. So they circulate, in a relatively direct way. But we didn't look in the blood.
Cool. All right, and the other question is from Tony Pan, who asks, how are you able to determine the optimal number of topics?
Oh, that's like the question for all machine learning. How are you able to determine the optimal anything? I will say 2 things on this. One is that there is often no single answer to that question. Just like in clustering or any other kind of analysis, different settings will actually give you different answers, and there can be multiple right answers, right? They give you different resolutions, programs that are expressed in different ways by different groups. So I don't think there's always one right answer. You can see sometimes you can identify when answers are definitely wrong. So you start to see like the same programs and extremely correlated gene sets getting split over and over again. And that's something that happens with non-negative matrix factorization methods a lot. They don't work great in sort of hierarchical settings unless they're really specialized to do that. And so we do look for things like that, over parameterization. But yeah, aside from ruling out very bad answers, I think it's not simple.
Sure, makes sense. All right, thanks so much, Sam.
Okay, thank you very much.
Right, so moving on to our last speaker. I'm sure the name is well recognized to most in this community, is Rahul Satija from the New York Genome Center, who's going to be talking about integrated analyses of single-cell data across technologies and modalities. Rahul, looking forward to your talk.
All right, hello everyone. So let me share my screen and you guys will let me know if there's any issues here. That looks promising. All right, so I'm going to talk about some very recent work that we just actually published a preprint on bioRxiv, and hopefully there'll be a link to the chat if you're watching on Workcast that you can use to follow along on that preprint. And the preprint describes some new technologies and computational methods. I'm going to keep this talk very light and not go into too much methodological detail. You can certainly follow along in the paper if you're interested in learning more. But we're really kind of excited by the overall vision and kind of path forward that these approaches sort of represent for multimodal analysis. And that's what I really want to focus on today. So, obviously you've heard a lot today and generally about technologies for single-cell RNA sequencing. And we often think about single-cell RNA sequencing as a way of defining our cells and what types of cells are present in our tissues. And that is certainly true. It's also interesting to think about flipping the problem around and thinking that what we're learning in single-cell RNA-seq is something about our genes and something about the patterns or the combinations of genes that can be co-expressed at the same time in any biological context. And of course, the role that any individual gene is playing might be different in one cell compared to another, and that's something that we're learning through these experiments. In the same way, when we do things like single-cell attack-seq, we're also defining our cells, but we're also learning something about the function and the role of each individual piece of DNA and how that varies from one cell to the next. And I think that's very exciting to think about learning sort of fundamental aspects or fundamental roles of pieces of DNA in the human genome from these types of single-cell technologies. And chromatin-based assays at single-cell resolution push us in that direction. But I've used this very sort of provocative word here, function. And I think single-cell attack-seq is a wonderful assay, but it tells us more about the accessibility or the openness of a piece of DNA rather than its individual function. So how can we start to explore the functional role of DNA elements in single cells? And one way of doing this, of course, is to look at histone modification. So as many of you or all of you know, the histone code hypothesis says that combinations of multiple post-transcriptional modifications on histone proteins are correlated with the relative function of an individual DNA element. So for example, the presence of the modification H3K4ME1 might indicate an enhancer. The presence of H3K27ME3 might indicate a repressor, but there's a variety of histone marks, which again, not just one at a time, but in combination can tell us something about what an individual piece of DNA is doing. And so there's obviously a lot of interest in being able to measure a combination of these histone modifications together in single cells. And that brings me to kind of a wish list for single cell profiling. And I'm gonna tell you about a technology called scDNA-seq. And I've named it that because it is not what I'm going to tell you about today. It does not exist. It's something we want, but it's something we haven't invented. If we could design some theoretical technology that could do anything we asked for, I would love to have a technology that could simultaneously profile the genomic location of multiple histone modifications in the same sample at the same time. I'd love for that technology to be able to scale down to single cell resolution. And while I'm wishing for things, I'd love to be able to measure all sorts of other molecular modalities at the same time, the transcriptome, protein levels, all at the same time, all in single cells in some giant multimodal assay. So that's something that we in many groups are working on, but at the moment that is far beyond the possibilities of what technology currently allows. But if we could do this, I would argue that it would open up a lot of really exciting analytical opportunities. For example, we could assign a functional role to every segment of DNA based on the combinatorial pattern of multiple histone marks. We could explore how that functional role changes across single cells in a heterogeneous population. We could identify DNA elements or motifs that correlate with these different functions. And we can learn how changes in chromatin state and function correlate with changes in gene expression and other molecular modalities downstream. So we could start to explore functional roles of DNA and also explore sort of co-regulatory relationships across modalities through multimodal data. So that would be really exciting if we could achieve this particular technological vision. And while we can't necessarily run scDNA-seq today, what I'll tell you about is something that I hope gets us towards a similar goal. And this has been work that's been led by two postdocs in the lab, Bigjie Zhang and Avi Srivastava. And their work really builds upon a series of advances over the last few years from the community. And I really want to highlight what those advances are before I tell you what we've developed. So the first advance is a technology called single cell cut and tag, which is developed by a number of labs, and in particular by Steve Henikoff's lab. And this is a way of being able to measure protein DNA interactions at single cell resolution. And many of you are familiar with cut and tag, but just in case you're not, you can think of it as very similar to attack-seq, But instead of having that transposase just integrate into accessible or open regions of the genome, that transposase is specifically targeted to regions which contain a particular histone modification or DNA protein interaction that's marked by an antibody. And it's using a sort of fusion transposase, it's targeted directly to these regions of the genome, which enables us to generate these genome-wide binding maps. The nice thing about using these transposases is they work very well on low input, including in single cell. And so earlier this year, a couple of labs generated the first single cell cut and tag data sets in both the mouse brain and also in human blood. And this is really exciting data. This is an example here from the mouse brain. And what you can see is we get a lot more information than just kind of openness or close or accessibility like we would from a TAC-seq. Based on these combinations of histone modifications, we can identify, for example, promoter regions based on the presence of H3K4ME3 that are active in some cell types, but not in others. We can look at actively transcribed regions based on different histone modifications. We can look at enhancers based on H3K4ME1, repressive regions as well. So it's really exciting data, particularly at single cell resolution. So this is a major advance, but there are some real challenges with single cell cut and tag. The first is that the data is extremely sparse. We're often dealing with hundreds of fragments per cell, which, compared to the size of the genome is very, very sparse. So that's not ideal for unsupervised cell clustering. And an even bigger challenge is when you're measuring these marks, as these technologies can do individually, one at a time, how can you integrate them together, especially if you're measuring an active mark like H3K4ME3 in one experiment and a heterochromatic mark like H3K9ME3 in another experiment? Trying to combine these different experiments together is an extremely difficult, if not impossible, computational challenge. So I'll tell you kind of how we've thought about overcoming that through new technology. And one of the fundamental ideas behind how we do this was introduced by the Technology Innovation Lab here at the New York Genome Center in collaboration with my lab and a technology that many of you know called CiteSeq. And what CiteSeq does is it measures RNA and protein simultaneously in single cell. It's a multimodal assay. And it does this by using antibodies, just like we use for flow cytometry, except they're conjugated to DNA oligos that we can read out alongside the cellular transcriptome, which enables us to have this sort of multimodal readout. So CiteSeq is a very powerful assay. Earlier this year, my group, in collaboration with the Technology Innovation Lab, published a very large data set using a huge antibody panel and a large number of cells, and using some new computational methods as well. We were able to generate sort of an atlas or a very high resolution set of cell states defined based on both RNA and protein in the immune system. We found this atlas very useful for interpreting new data sets as well. So that was one particular method that was useful for us. And then last year, or actually earlier this year, the Innovation Lab extended CiteSeq into new technologies, ASAP-seq and DogmaSeq. And what those technologies do is they also measure cell surface proteins, but they measure them in combination with chromatin state, with a TAC-seq. So you can measure, for example, a TAC-seq and cell surface proteins at the same time. So it's a really cool method, and you can read more about this. I want to highlight that one of the key advances that makes ASAP-seq possible is the ability to perform a TAC-seq on whole cells rather than just whole nuclei, because of course we need to be able to capture the surface proteins as well. So there's a series of technologies that have come out that we've built upon, and what we're introducing here and what I'll tell you about today is a technology called SC Cut and Tag Pro. And so this is a technology that enables us to measure cut and tag profiles for a single histone modification or DNA binding protein, combined with a panel of cell surface proteins at the same time. So the way this experiment is going to work is we're going to stain our cells with antibodies against our cell surface proteins of interest. So we have a large antibody panel that we use. We also stain cells with an antibody against our sort of cut and tag target of interest, for example, the histone modification H3K4ME3. We then run the 10x single cell ATAC-seq kit with optimized conditions that enable us to run on whole cells rather than whole nuclei. And importantly, we use the TN5 protein A fusion transposase that directs that transposase complex to regions that are marked by our histone modification of interest. And as I said, at the end, we get multimodal data. We get a cut and tag profile for a single modification and cell surface protein levels at the same time. So I'll show you, we've generated quite a lot of data in human PBMC, and I'll show you a lot of that data today. What I'm showing you now are what happens if we cluster the data for, these are 3 representative marks that I'm showing you, based either on the cut and tag data or on the protein data. Although they are measured in the same cells, we've just split them artificially here. And what you can see is that if we cluster based on cut and tag, we are able to discover cell states in the immune system, but it's very sort of low resolution annotation. That's due primarily to the sparsity of cut and tag data. On the other hand, if we use our protein data that's measured in the same cells, we can get very high resolution and very highly accurate cell annotations by using these cell surface proteins. But even more importantly, the measurement of these cell surface proteins serves an incredibly important focus for us, which is it enables us to integrate all of these different data sets together. So over the last few years, as I've mentioned, we've accumulated quite a lot of data in the human immune system. We've accumulated a huge amount of site-seq data where we've measured RNA and protein together. We've accumulated ASAP-seq data where we measured attack-seq and protein together. And then we've collected all of the single-cell cut and tag pro data where we've measured these histone modifications in Pol2 binding. And again, we've measured protein in those experiments as well. So in each individual experiment, we're measuring a different modality, but we're always coupling that measurement with the same panel or a very similar panel of cell surface proteins. And what that enables us to do is very seamlessly integrate all of these data sets together based on the protein information. And that we think is extremely, extremely powerful. So what I'm showing you now is all of the data that we've collected actually over the last few years in human PBMC, featuring 10 different molecular modalities, all of which have been placed into the same kind of harmonized and well-integrated space. In addition to visualizing these data sets together, we can also annotate the cells very consistently using a high-resolution set of cell labels. That's what I'm showing you here, regardless of which technology or which modality was being measured. And in fact, using a series of computational tricks that we go into in the manuscript, we can actually create synthetic profiles of individual cells where we've either measured or predicted all 10 of the modalities simultaneously. So we refer to this jokingly as single-cell mega-omics. To be clear, we didn't measure all of these histone modifications and chromatin accessibility and RNA and protein and Pol2 binding all in the same cells. We measured them in different experiments. But because we're able to integrate all these data sets together, we can kind of get the next best thing, which are very high resolution and well-integrated predictions of what a cell would have looked like had we measured everything together and simultaneously. So I'll just point out that our single-cell cut and tag profiles are very consistent with ChIP-seq data sets that have been generated, for example, as part of the ENCODE project. In the ENCODE project, they took human PBMC, they sorted cells into different groups and then did bulk experiments. For us, this is all sort of pseudo-bulk tracks that are generated from our clusters that were determined at single-cell resolution. So it's nice to see that we can generate high-quality cut and tag profiles for all of these different histone modifications and all of the cell types that are present in human blood. And again, I'll emphasize that it's really fun. It's been kind of a goal of ours for a while to be able to have all of these modalities measured together in exactly the same set of cell types in a way that allows us to compare how the modalities are behaving across different levels of the central dogma, all at the same locus. So for this is the CD8 locus, which is, of course, expressed in CD8 T cells. And what you can see is that this gene is, of course, expressed primarily at the RNA and protein level in CD8 T cells. It's also uniquely accessible in CD8 T cells. It's closed everywhere else. You can see a variety of activating histone modifications that are present in CD8 T cells, both for promoters and also for enhancers. But then in the other cell types, you can see that the gene isn't just, not only is it transcriptionally silent, it's also actually being actively repressed. We can see high levels of this repressive modification, H3K27ME3. So yeah, it's fun to be able to see all of this together, all in the same set of cells. So the question now is, can we go back to this original goal? Can we define functional states in every bin of the genome using this combination of multiple histone modifications? And there are tools for being able to define chromatin state based on histone modification data. The most popular is a tool known as ChromHMM from Jason Ernst and Manolis Kellis. And what we've done in this manuscript, and I won't go into the computational details now, is we've extended this tool into something we call single-cell ChromHMM. And so the way single-cell ChromHMM works is it first uses the Baum-Welsh algorithm to determine an overall possible set of functional chromatin states that exist based on our combinations and multiple histone modifications. But then it goes back into individual cells and individual bins of the genome, 200 base pair windows, and it probabilistically assigns each of those windows to one of the functional states in each cell. I want to highlight that single-cell ChromHMM does not require a fixed set of cell labels. It works at single-cell resolution, so it's particularly well suited for cases where you have continuous cellular phenotypes, like you've heard about earlier today, like trajectories, or cases where it's not exactly clear what is the right level of resolution to discretely cluster cell types. So the details of this are really in the manuscript, but I'll just highlight some of the results. What I'm showing you here are some of the different states that chromatin M and single-cell chromatin M can characterize. So we discover very clear promoter states that are marked by open chromatin accessibility and K4ME3. Similarly, we see active enhancer states as well. We see a very strong repressive state marked by K27ME3 and no chromatin accessibility. And we also see a very strong heterochromatic state marked by K9ME3. And then, of course, large portions of the genome are unmarked and don't have any chromatin modifications as well. So as I mentioned, the output of chromHMM is that in each individual cell, for each 200 base pair window, we get a probabilistic classification of what we think that window is doing in that cell. So here's an example. This is the PAX5 locus, which is expressed particularly in B cells. And you can see here, this is a window that's located at the transcriptional start site, this yellow window here. You can see that in each one of our B cells, this window is annotated as high probability at being in a promoter state, which is because we see a lot of H3K4ME3. So we know that's correct because this is the transcriptional start site. In all of the other cell types, we see a very high probability of repression, again, demarcated by H3K27ME3. But we get this for every window of the genome. So if we scan over a little bit here, now we can look at a region that's an enhancer. And again, now in every B cell, this region is marked as an enhancer. But in all other cell types, the region is very strongly repressed. So in fact, you can even make some fun movies. We can scroll across the genome and we can see the switch from an enhancer into a promoter state while other cell types remain repressed. So this is a lot of data. At every window of the genome in every cell, we have some idea of what we think that window is doing. So as I mentioned, one of the fun things about this is that it really enables us to explore how the function of an individual DNA element changes across, for example, a developmental trajectory, because single-cell chromatin M is really well suited for continuous analysis. So we're very excited to apply this to more transitioning systems, in particular developing systems. But for now, we started by looking at a trajectory that describes sort of T-cell maturation. Starting from naive cells, moving towards memory and eventually effector state. So we're looking at CD8 T cells here, and we can define a single trajectory across all of our integrated data sets that describes the progression of cells along this maturation state. And then we can go back and we can ask how many regions of DNA are changing in their function over the course of this trajectory. So that's what I'm showing you here. Every window in this heat map, every column, in this heat map is a cell. Every row is a genomic window, and I'm visualizing the probability that that window in that cell is in a repressive functional state. So for example, at the top of this heat map, we're visualizing windows that start in a repressive state in CD8 naive cells, but lose that repressive state over time. Whereas the bottom, we have windows that gain that repressive state over the course of maturation. And I would say that one of the things that surprised us is that there's an incredible global rewiring of chromatin during this transition. There's more than 15,000 genomic windows that change in their chromatin state over the course of this trajectory analysis. By contrast, there's only a few hundred genes that change in their expression, but there's a much wider a set of regions that change in their chromatin state. We can also identify particular regions or motifs that are associated with these regions. So for example, these are very well-characterized regulators, TBX21 and LEF1. Regions that have a TBX21 motif often start this process in one chromatin state, but end this process in another chromatin state. So this is a very interesting way for us to be able to nominate regulators that are associated with cellular transitions as well. And I would say that one of the kind of fun outcomes for this and sort of questions that it raised was just how variable is repressive chromatin, even in this population of fully differentiated cells. We know that chromatin and gene repression through K27ME3 is very important in the early embryo, for example, but we were surprised to see so many changes in a fully differentiated system like human PBMC. In fact, as you know, we can cluster cells based on their transcriptome or based on their sort of chromatin accessibility. We can also cluster cells based on their genome-wide probabilities based from single-cell chromatin based on their posterior probabilities of repression. And to our surprise, we get almost exactly the same clustering with exactly the same level of resolution. And so what that tells us is that the identity of a cell is not only deeply encoded in the genes that it chooses to express, which of course we know, it's also deeply encoded in the genes that it chooses to actively repress, which we think is kind of a fun and kind of interesting result. It then leads to the question of, well, what are the genes that each cell chooses to repress and why is it repressing them? So in some cases, the answer is obvious. So I showed you before, for example, the CD8 locus. And the CD8 locus is a very sort of clean and internally consistent example. So it's a gene that we know is expressed and functionally important only in CD8 T cells. So it makes sense that all of the active modalities and all of the active histomodifications are present in CD8 T cells, and the repressive modalities are present in other cell types. So, I mean, you know, it's cool to be able to see all of this together, but it's not particularly surprising. Everything is kind of telling us a very consistent story. But it turns out that the CD8 locus is actually more of a rare case than it is a common rule. Far more frequently, we see cases like the ones that I'm showing you here. So these are 3 loci that exhibit incredible heterogeneity in their chromatin state. So you can see here, for example, that in monocytes, the DUS5 gene is being very actively marked by this repressive modification K27ME3, the same with the other 2 loci. So there's substantial heterogeneity in chromatin state. What's surprising to us, though, is that we see absolutely no difference in the expression of the RNA for this individual gene. And the reason for that is because the gene is basically shut off in all cell types. So it raises a really interesting question. Well, I think there are 2 things that are interesting here. One is, this is a locus that from the perspective of defining cell types is highly informative when looking at chromatin data, but not informative at all when looking at RNA-seq, because the gene is just off. So there's no information there, but there's a lot of information in the histomodifications. But the other thing that's interesting is, you know, why have monocytes gone through the trouble of actively repressing this gene when it was transcriptionally silent anyway? The gene is, even when you lose K27ME3 repression, the gene isn't on. So why is the cell going through the effort of trying to actively repress this gene? I think this is kind of an interesting sort of philosophical question. I'll note that we see hundreds, if not thousands, of these examples. In every case, when we see this, it's because the gene is basically transcriptionally silent across the genome, across cell types. And for whatever reason, individual cell types, even though the gene is silent, are making an extra step of repressing this gene actively. So one really intriguing hypothesis from why this might be taking place is that this repression isn't really changing anything about the steady state of the cell, but it might be encoding something about the future potential for what the cell could do. So maybe the cell will encounter some sort of environmental stimulus or some sort of future exposure. And when that happens, in some cell types, the gene will be competent and be capable of being turned on, whereas in other cell types, that active repression will prevent that activation. Indeed, we do see that these genes are very heavily enriched, at least from the perspective of Go ontology, for at least interesting categories. We see a lot of transcription factors, signaling proteins, ion channels, things which we would expect to be differentially expressed upon a new stimulus. But I'll say that this is an open question for us. We don't know the answer yet, and it's something we're excited to explore over time. We're also excited to explore the developmental dynamics for these loci and to understand when in differentiation and development, for example, monocytes chose to actively repress this gene. The very last thing that I want to show you in the last couple of minutes is, I think, another fun application and sort of possibility that we can accomplish with our multimodal atlas. So as I mentioned, you know, we've accomplished, or we've assembled this very highly multimodal atlas that consists of many, many different measurements all in a harmonized space. And we were able to do that because in each experiment, we measured all of these cell surface proteins. But what about cases where you don't measure cell surface proteins? So for example, what if you're working with nuclei? So as I mentioned, you know, earlier this year, the Hennekoff lab published a single cell cut and tag data set on nuclei. So there was no accompanying cell surface protein measurements. And it's very nice data. You can see a variety of cell types that are emerging in the immune system, but it's not the same level of resolution that we see in our multimodal atlas. And so our question was, since in our multimodal atlas, we kind of know what we're looking for, can we use this information to help interpret a new query data set like we're seeing here? And in fact, this works extremely well. We can take our query data set, even though we didn't measure proteins, and we can map it onto our multimodal reference. And when we do that mapping, now all of a sudden we can see, even though it's exactly the same data, I'm showing this the same data on these 2 plots. The left is unsupervised analysis. The right is what happens when we map onto our reference. And when we map onto our reference, now all of a sudden we can see substantial heterogeneity within monocytes, B cells, multiple subpopulations of T and NK cells. We can substantially increase our resolution of cell type annotation because we know what we're looking for. And so what that tells us is that in the future, whenever we're generating new data sets from human blood, we can use this multimodal atlas to really improve our ability to interpret and analyze that query as well. All right, so I'll just stop there and summarize a few things that I've sort of fun things that we can do with these approaches. We're really excited to be able to explore chromatin remodeling during cellular transitions using single cell cut and tag pro and also single cell chrome-HMM. We can identify genomic regions whose regulatory function is dynamic across a developmental trajectory, and we can specify DNA motifs and factors which may regulate these transitions. We're really interested in this idea that repressive chromatin encodes substantial regulatory priming, and that not only is a cell state fully encoded based on its repressive chromatin landscape, but also many heterogeneous loci at the chromatin level are not associated with RNA changes at present time, but they may be associated with RNA changes in the future. And lastly, the ability to assemble this mega-omic atlas creates a very unique reference for the projection and interpretation of new data sets, and substantially improves the granularity of cell type annotation for new single cell cut and tag data sets, even if no proteins were measured. So I'll wrap up there. I want to thank Bingji and Avi, who have really led all of this work. And I want to acknowledge a very long-standing collaboration between my lab and the Technology Innovation Lab at the New York Genome Center, as we've worked together over the past 5 years to develop both multimodal technologies and multimodal analysis strategies. So with that, thanks very much for listening, and I'm happy to take some questions.
All right, thanks so much, Rahul. It was a terrific talk. Actually, one clarifying question for me before we get to a few of the others is, were you actually using oligo-conjugated versions of the actual histone targeting antibodies like in this? Are they part of the site-seq modality that you get out of this?
No, so the histone targets themselves, those are just used to recruit the protein ATN5 enzyme or transposase to the right regions. Those are not something that we read out in the same way that we read out the site-seq proteins.
I'm curious, do you think there would be any value in doing that? Like maybe even from like a normalization perspective of like total quantification of things?
Yeah, it's a really interesting idea. There's been a lot of really interesting work. I think it's from Long Kai's lab, maybe it's not Long's lab, but a variety of labs that sort of use the overall amount of different histone modifications within a cell as an interesting level of cellular phenotype, not necessarily localized to individual regions of the genome, but just sort of the overall amount of histone modification changes quite a bit during dynamic processes. So it's an interesting idea. We hadn't thought of it.
All right, so a few questions. One from Anil Raj from Calico Life Sciences, who asks, can you say a bit about how you get the correlation between a pair of modalities, for example, H3K27 acetylation, H3K34 methylation, given the only joint information between each modality and the surface antibody? So essentially, do you assume that conditional on a specific antibody modalities of interest are sort of independent of each other?
I don't think we assume anything about the relationship between the modalities, and I think that's really important. So, if you followed my lab's work, you know that for a while, we were trying to integrate RNA-seq and ATAC-seq data sets together by making some assumptions. For example, we would assume that if a loci was open at the ATAC level, it was probably being transcribed. And those are some tough assumptions to make. They may hold sometimes or more often than not, but they're going to be many counterexamples. So what's nice about these multimodal approaches is we're not making those assumptions. We are assuming that cellular state can be defined by the proteins that we measure. If the proteins were not informative, this wouldn't be a very useful assay. If the proteins are informative, we can basically use that combination of protein to assign very high resolution cell state, both at the discrete and at the continuous level. And then we can match cells from different experiments based on those antibodies. Once we've done that matching, now we can go back and we can look for cells that are in a similar biological state. How do different modalities relate to each other? And so I like that approach because I think it reduces the number of computational assumptions that we're making.
Cool. Thank you. So another question from Zipeng Mu, which was that Big recalls that there's a discussion among people doing cut and tag that suppressive markers are hard to measure because the chromatin around suppressive markers is not as open. So the TN5 has a hard time accessing those regions. So he's wondering if the protocol used in ST cut and tag pro is different between active and suppressive markers.
Yeah, it's a really great question, and it's something that we remain a little bit puzzled by. But I will say this: we can detect very, very strong peaks for K27ME3 and K9ME3, which are very strongly repressive marks, even when there is no ATAC-seq signal. And so what that tells us is that it's certainly possible for the fusion transposase to be recruited to these regions of the genome, even if the chromatin isn't at least accessible from the purposes of ATAC-seq. We also see a very high correlation between our data and ENCODE ChIP-seq data, which suggests that there's no massive systematic bias that's preventing us from being able to look at these regions. It does raise a really interesting question of why are these, if the chromatin is closed, I guess the pull from the antibody is strong enough that it will still enable the transposase to enter. It kind of makes you wonder why the transposase doesn't enter without the antibody. Maybe it sort of prefers, I know, it's a really interesting question. And I can't say I fully understand why it works, but I do think it does work.
Cool. And a final question that I might expand out a bit. So specifically, the question from Kwan Nguyen was that, how often do you see the CD4 locus expressed in a CD8 T cell across the CD8 T cell differentiation trajectory? But I wonder if maybe a broader question there is, if you can comment on the relative importance of sort of repression versus not in terms of what the ultimate consequence on gene expression is. Like, are there any sort of generalized patterns emerging there, or is it really specific to individual loci? Yeah,
I mean, this is a fascinating question to us. I will just say my initial thoughts before we generated any data. I mean, obviously, K27ME3 or polycomb-mediated repression is an extremely important mechanism for gene regulation. But I didn't think that would be a very active method of encoding cell type specificity in human PBMC. I would have thought that it played a very important role in the early embryo and maybe in hematopoiesis, but that by the time you had reached kind of differentiated cell types, you're obviously going to have genes that are on and genes that are off, but I didn't expect to see so many differences in terms of polycomb repression. So in terms of the relative importance of these modalities, it's definitely changed. My prior going in would have been that the vast majority of regulation was encoded at the level of activity. And I don't now. I'm much more open to thinking about how that changes going forward. I will say that a fun conceptual idea, and this is really just kind of this would be fun, I don't know if it's true, is that this repression is encoding something about the future potential of a cell, and that especially thinking about what a cell could do going forward, this is really something that we just weren't able to measure before. As I said, it doesn't even show up in a TAC-seq, so it's great for us to be able to measure this. But we have to prove that, and I think that's something we're going to enjoy exploring for the next few years.
Cool, great. Thanks so much. So I think with that, we're going to move on to sort of our moderated discussion section. So if I could ask Samantha and Sara to come back into the studio, if we can get this to work.
Yeah.
There we are. All right. Cool. So actually, just to kick this off, I just want to catch up on a question that was asked earlier that we didn't get to, which was from Meredith Singer, and I think the question was more directed probably at Samantha, which was, how do you see the advantage or disadvantage of the total topic weight per cell being 1? Specifically, the normalization of the cellular topic weight to add to 1 could show different results than in an unnormalized setting. So love to hear any thoughts you have about the need or the danger associated with the add to 1 requirement.
So I think she's talking about the fact that topic models use probability distributions. And so both, unlike other kinds of non-negative matrix factorizations or other kinds of latent factor models, you really have to think about the result as a distribution. I think for me, I hesitate to I feel like I don't have enough experience either on the theoretical side or in the actual application side to really generalize about potential dangers or anything like that. But I think, the sort of rule of thumb has been that actually aids in interpretability. And I would say that's actually been very true for us. Of course, it adds constraints that aren't necessarily, you have to be aware of them when you're actually interpreting the results. But if you think about how we often take any kind of data, we often normalize it so that things kind of have the same denominator. And it's an intuitive kind of way of being able to understand, weights as being sort of relative. And that becomes really hard if you can just sort of add infinite quantities to any coordinate. So I think there's also a way in which the Dirichlet prior and I'm probably not saying that right. I think it depends if you're anyway. Whatever. Say that the prior and the probability distribution together also kind of enforce a sort of sparsity, not the kind of sparsity that you get from like an L1 type penalty, but I think they do. That also aids in the sort of interpretability that you get from these. And in general, we've just, we found that just kind of optimizing the Frobenius norm match, which is what NMF does, it's very difficult to actually use the results in the same way that we've been able to here. There are ways of kind of correcting those and fitting them better, but I think probably the ideal thing is to sort of change other models that are maybe more efficient so that they have some of these properties as well.
So I think another question that was originally directed to you as well, or probably in response to your talk, but I think it's actually it's one of these it's always sort of the bomb that's thrown at all the Human Cell Atlas meetings on this topic of like cell states. But I think or cell types and states. I think it's
relevant for everybody.
Yeah. I think an interesting nuance here is from Paul Maurizio at the University of Chicago.
Can anybody else hear him? We've lost you.
Yeah,
sorry.
Type your question.
Somebody muted us.
I was just chatting there for no reason. Yes. All right. What was
so what was the question from the University of Chicago?
It was to what extent are continuous immune cell states representative of new underlying biological functions versus byproducts of reduced resolution between functional types so the question is specifically at the RNA level compared with traditional classification of protein level, but I think even in general, so sort of maybe how much is a continuous state truly continuous, and maybe historical discrete measurements, while they may have been less resolved, may actually represent more distinct sort of functional states at the end. So Samantha, I'll put that to you first, and then maybe.
I'm gonna get in trouble with all the immunologists now. So this is, yeah. I hope maybe there's not that many listening to machine learning and immunology talks. I'm like firmly on the side of there being a lot more intermediate and continuous kind of phenotypes than are allowed for by traditional immunology models. I don't think that means that the traditional models aren't helpful frameworks and don't capture a lot of important phenomena. That's clearly true. But I just think like it's sort of happening in every direction all the time that we see actual functional behavior from cells that have mixed effector phenotypes that have combinations of transcription factors or cytokines that we don't expect, that respond to cues we don't expect them to respond to. It just, I think though, it's real. I was actually thinking about how it's really nice the way that fits into kind of Sara and Rahul's work, that I think we don't really understand the mechanisms of control very well, and I'm not even getting into those, but I think that these phenotypes are reflected at the chromatin level. You can actually see potential for multiple states even at the chromatin level in cell types, and we just haven't been able to really look at them or understand their significance. By the time we would see them normally, they would be really sort of in the more extreme state. So I think our understanding comes from what we've been able to measure. And also the other thing I want to say is just from the point of view of experimentation, in vitro models tend to cause very extreme phenotypes. And so the more that we've been able to look in vivo, we tend to see way more confusing phenomena than when we kind of push cells to their extreme with using in vitro models. So I think that's another reason why we're getting a more sort of complex picture.
Thank you.
Maybe somebody else has an idea about this and could join me in pissing off immunologists everywhere. I don't know.
Well, you're going to have an immunologist asking you a question now, or two questions. Yeah. First, I want to thank everyone for fabulous presentation. Very exciting. Samantha, you mentioned IL-5, yes, right, in the development of the IL-6s. Have you had a chance to do an experiment with IL-5 knockout mice that would have given you the solution, whether it has a key role or not in that?
Say that one more time. Sorry, it was a little quiet.
Okay, you mentioned IL-5 in the development of your ILC2 to ILC3 transition. It would be good to do an experiment with an IL-5 knockout mice. Have you had the chance to do that experiment so that to ascertain the role of this IL-5?
So your hypothesis would be that somehow, sorry, my camera is going all over the place. I like to see the person I'm talking to, and it keeps showing me random other people. Yeah, exactly. It's like shifting all the time who I'm looking at. I find it distracting. So you're suggesting that that the IL-5 signaling would be important for that transition.
Yes,
is that's
what I'm wondering.
Yeah, so that's an interesting possibility. So what we know, no, we didn't do it. I don't think we did this on an IL-5 knockout. We had, just the fate mapped IL-5 mouse. And based on that, what it looks like is that cells both that go through the kind of effector ILC2 state where they express IL-5 and then don't, as well as cells that never go through that, both end up becoming ILC3-like. Okay. But that doesn't say that it would still happen without it. I mean, that's my guess based on what I've seen in the biology of the ILCs is that you don't need IL-5 signaling to have that happen. But it's an interesting question. It would be really hard to piece apart sort of the ILCs. It's possible that the ILCs that are sitting there at steady state would be different if they didn't have, IL-5 signaling and might respond to different cues. So it's an interesting question. I would suspect that you would not require it, but I can't be sure of that. And we didn't do that experiment.
And to give you even a harder time, I mean,
sure.
Do we have? I
asked for it.
The same kind of information in the human. Have you been able to look in the human skin and see whether you have those ILCs, this arc of ILCs? I mean, I think this is very interesting.
Yeah, so humans are interesting for lots of reasons. So we definitely, we didn't get to look in them, although we would really like to, but we have had, we've had collaborators who have. So we definitely see ILC3s in human lesions, and you definitely see ILC2-like cells in human skin. And another thing that was recent, I'm trying to remember, I'm blanking on the lab's name, but there was really interesting work that showed the existence of sort of precursor-like cells in the skin that could be, sort of progenitor ILC type cells, and they look kind of vaguely like our quiescent ILCs that we see. They have some of the same phenotypes, and some things are different. So I think there's a strong suggestion that some of the same biology is true, but there's also been more reports of circulatory ILCs being relevant in skin inflammation in humans. So there may be additional mechanisms.
Thank you very much.
Well,
that wasn't so hard.
Well, we tried not to be embarrassed.
Well, so I just give you a break. A question for Sara, which is that most of the work that you presented in your talk was really on kind of an ATAC-seq from InGen, which is really on these sorted populations, and you mentioned kind of the plan to move into the single cell ATAC-seq. I'm curious. I don't know how far you've gotten into that work already, but just on balance, or how do you view the balance between the relative loss of signal due to the sparsity in the single cell ATAC measurements versus the sort of increased number of measurements now that you're using kind of the cell as the unit as opposed to like a sample as a unit?
Yeah, that's a really good question. So there's a few points that I wanted to make. So one is that it's actually pretty challenging. And so initially what I thought you could kind of try to answer what's the right resolution by actually figuring that computationally based on how well you can make prediction at various granularity levels. And so this is kind of the line of more on the methodological line that we've been pursuing. Can we figure out computationally at which kind of granularity we saturate on predictions? And kind of going through this exercise, and I think it really depends on sequencing depth. But it seems like, like some sort of meta cell resolution where at least something like 50 or 100 cell information from 50 to 100 cells combined to define a unit, that seems to be most stable for computational predictions of this problem. But yeah, I think this question of resolution is really interesting, and there is a biological angle to thinking about it, and there's a computational angle to thinking about it. And that's kind of on the computational side, I have a lot of thoughts of how we can use models to actually decide with the current data that we can generate, what's the resolution that we can support modeling them and actually figure out stable models that make accurate predictions.
I'm curious, maybe this is I guess I'll go the question to you first and then also into Rahul, which is the addition of additional modalities such as ASAP-seq obviously brings this additional dimension that might help to reduce noise or provide sort of additional information about restricting what that metacell space might look like. So it's not just unbiased. Like, have you thought of incorporating any of that into your work?
Is it for me or Rahul?
I guess for you originally first, I guess. It's like, we know Rahul's incorporating his, so.
Yeah, definitely. I think those are definitely really interesting directions to kind of have some sense of what are the possible states or some prior about what the possible states are. We haven't really gone that direction because I like this idea of being really unbiased and then discovering what the data has to say. But I also see like the benefit of the other side. So it's just like kind of balancing two different desires.
Rahul, do you want to provide your perspective on that same question?
Yeah, I mean, I think from the idea that there is I think one of the interesting things about what I showed is we always measure these proteins. We barely discuss any of the results or any biological findings that come out of measuring the proteins. We're just using the proteins as a source of integration. For sure, there's plenty of interesting, we can find markers and so on for facts, and it's not that we can't do that, but even if we didn't care about the protein levels at all, they wind up being phenomenally useful for defining cell state, enabling us to integrate these together, which then allows us to focus on chromatin state, which we are really interested in. Mm-hmm. So I think that idea of measuring something for the purposes of integration is a very nice experimental design, because we want to measure all of these things. We want to explore relationships between many different modalities, but we cannot measure them all at exactly the same time. And once we can, we'll come up with new modalities that we then can't also measure at the same time. So I think this approach of sort of trying to bridge it together with a series of multiple experiments, but where there's always something in common that facilitates high resolution integration and also very accurate integration, that's a strong approach from our perspective. It doesn't have to be protein. You can measure RNA and histone modifications, RNA and attack, RNA and protein, and anchor together based on RNA. You can anchor together based on attack. There's all sorts of ways of thinking about this, but I think the general strategy is one that we're excited about.
Rahul, at this level, I'd like to ask you a question, very practical questions for an immunologist. We have done with colleagues, Dr. Pasquale, for instance, a lot of staining of patients with lupus. We have samples we have done a couple of years ago, and now you are telling us that, well, you don't need to put your batches, so to speak. You can analyze data with different modality at different time points. The practical things, can I go back to my frozen sample, do the experiments, and then with the tools you have developed, be able to say, what I found 20 years ago, now I can explain it? Or should that be rather done, those experiments, should they be rather done at the same time or under a condition that you control better?
So I think there are two questions there. One is sort of the potential for reference-based analysis in particular to be able to improve the interpretation of low-quality data. And this, I definitely think, is something that is very, very powerful, and we strongly recommend. So many of you, or some of you, may be familiar with our Azimuth toolkit, where you can go to a website. In fact, Jacques, we've used your samples, as you know, to demonstrate this. You can upload your counts matrix, and it will automatically map data onto our reference. And we find that particularly when you have shallow cells or cells with low-quality RNA, or cases where the data quality is less than ideal, which can prohibit high-resolution annotation from unsupervised clustering, knowing what you're looking for is very helpful. And a very simple analogy is that when we assembled the human genome you needed very long reads. You needed 500 to 750 base pair reads. And now you can get away with 25 base pair reads. If you had shown a 25 base pair read to somebody, in 2001, they would have thought of it as like a cell with like 50 UMI. So we can make sense of low quality data when we have references. So I think that's very valuable for being able to go back into biobanks and cases where samples are not necessarily high quality and interpreting them. The second question is, what do you compare those samples to? Can you compare, if you want to know what's special about your lupus sample, can you compare that to our reference, which we generated last year? Or should you actually just, should you also run samples from healthy controls that were processed in exactly the same way? And I think from the perspective of experimental design, you should it's always better to be able to match your experimental design for your cases and controls as closely as possible. And so we would encourage you to, you could also upload your control data to the same reference and map it, but then you're always comparing control and cases that you yourself generated under similar conditions. So that's a kind of idealized answer. Sometimes in practice, you may not have matched control data sets. In blood, you often can get matched control data sets, but in other tissues, people don't always part with, their bone marrow or their spleens. Then it's a really interesting question about what do you compare it to? But I would argue strongly that question is not unique to single cell analysis. That's a general question for how we do comparative analysis when we can't have an ideal experimental design.
I agree. Thank you very much.
Cool. So maybe just to end on a couple of sort of philosophical questions for the group, maybe. Well, actually,
Can I add one? Can I add one thing to what Rahul said?
Yeah, go ahead.
Rahul, I love that analogy, and I was just thinking about how, if you wanted to take it one step farther, it's like, we had the reference genome, which allowed us to use shorter reads, but we still probably didn't understand anything about the variation that was out there because we had like one reference genome, and now we have many, many more reference genomes. So I think there's also that aspect that as we get more Alice's, more references and more contexts, we understand also the variation, and we have a better chance of even, there's that next level of sort of trying to understand where in that space, a sample fits in. Anyway, I really like that. I hadn't heard that analogy.
Yeah.
It did remind me of, it's unrelated, but it reminded me of another question I had to ask you, sort of, Samantha. It stems, I guess, from a thing that you showed. So I really like the validation that you showed at the very end there with the fate tracing mouse was really nice. But it also kind of ties a little bit into something Sara said about how there's a lot of noise and like a little bit of signal. So I think when we think about applying ML to immunology in general, like the general strategy is lots and lots of data kind of goes in, and it's in a somewhat unbiased way, and we hope that we have something that comes out, and then we kind of think of validation as a secondary step that happens afterwards to validate like a very specific ML insight. But how much do you think about like taking the results of that validation and using it ultimately to improve models? Like the validation, instead of being an endpoint, is part of an like iterative cycle. Is that something that you? Maybe I'll put the question to each of you, maybe starting with you, Samantha. Like, did you take the results of that fate mapping and reincorporate it into your models?
So let's see. First of all, my absolute ideal way of doing any kind of research in this space is to be iterative with experimental validations and perturbations. I'm guessing probably all of us would say that. Like the best way to learn things is to be able to get feedback, right? It's like a one-way conversation. It's very hard to make sure you're actually communicating what you think, and same thing in terms of learning. Right. So given that, it's also at this point, CRISPR is helping in some contexts, but I would say we're still a long way from being able to kind of do the kinds of perturbations that are really meaningful for immunologists, to like, they're very careful and very laborious, and they really want to see the specific mechanisms. And for that, you can really only prioritize a few kinds of things. So I actually see tons of stuff in that data set that I would love to pursue. But, I like all of that are sort of years of experiments at this point. So I think being able to both kind of speed up those kinds of validations, that would be really helpful. And also, figure out how to better prioritize which things are worth spending energy on, and then trying to, match the people who care about those kind of mechanisms to do the experiments. I think that's actually, there's kind of it's a web, right? But anyway, in terms of this specific setting, what I wanted to say is that I have used this intensely. And it's really a balance between overfitting, right? Like I don't want to like assume that this represents concepts that generalize too much, but I also feel like I learned a lot from this data set and from this whole setting about things that we're not good at yet. And I think those things translate to multimodal data, they translate to other modes. And so I think it's extremely useful to have even some small validations of kind of unexpected phenomenon, because it helps us kind of figure out what we are able to do, what we're able to learn, and what we're not able to learn. And I'm trying really hard to build on that without somehow overfitting.
I guess maybe that, since we're almost out of time, I just put that same question to Sara and Rahul as well, for your thoughts.
Yeah, I really appreciate what Samantha just said. So I think, computer scientists or like people that care about models a lot really use data to understand models, because we don't understand all aspects of the models. And like to stereotype the other way, biologists use models to understand data. And I think if we kind of look like basically if you have two people with the different expertise working on the same problem, then when we understand models and we understand the data through that model, and we can push the boundaries of both.
Cool. Well, I guess we are at the hour, so that's probably a reasonable point to wrap up at. I want to end with conveying one last sort of a comment, not a question from the chat, directed at all, which is just to thank you all and note that we appreciate your direction and inspiration in this area and guiding the field forward. So thank you so much for taking time to join us today, and also to all our listeners for joining as well.
Yep.
It was really fun. Thank you.
Thanks a lot.
Thank you so much.
Goodbye, everyone.
Thanks, everyone.
Enjoy your day.